My name is Hyojin Kim, working at Uniklinik Aachen in Germany. In this video, I'm going to explain the single-cell RNA-Seq data analysis from mouse renal fibrosis. The goal of this project is to interpret the pathology of renal fibrosis in the perivascular niche, the area surrounded by the blood vessels. So we focus on the cells where GLI1 or NG2 or MYH11 or PTG beta is co-expressed with TD tomato gene, because those four genes have been known to be expressed around the perivascular niche. So the biologists sort such cells with the expression of TD tomato. I had the 8 different data sets from the 4 different TD tomato data and the 2 different conditions. I also had the CD31 positive data set in order to focus on the endothelial cells. I performed the basic preprocessing, and the biologist labeled each cluster to the cell type, and I integrated the normal mice and the renal fibrosis mice for each TD tomato data set. So I integrated 2 conditions from the TD tomato GLI1, NG2, MYH11, PTG beta, and CD31 separately. So I had the 5 different integrated data set. This is the output of the UMAP from the integrated data set with the cell notation. The blue is the fibroblast, green is myofibroblast. The pink is pericyte, yellow is neuron. In the case of the CD31, it has different cell notations because it has the subset type of endothelial cells like glomerular endothelial cell and medullary endothelial cells. For the 5 different integrated data set, I performed the 5 different studies to infer the pathway activities, transcription factor activities, intercellular communication, gene set enrichment test, and the cell differentiation. I will explain these 3 parts together as functional studies. I visualized intercellular communication between 2 single cells or 2 cell populations. These intercellular communications could be connected to the biological pathways inside a cell, and each biological pathways could be connected to the transcription factors. So I tried to find the 1 nicely connected biological stories from the cell talk to pathway to transcription factor. I found the 6 biological stories, and these stories have been described in other studies. Here, I will explain one of these. I used the cell chat for intercellular communication, and this is one of the output from the cell chat. I found that the semastry signaling was more upregulated across the cells of the renal fibrosis mice than the normal mice. I checked which ligand and receptors contribute to these signalings, and I focus on the semaphorin 3F and the semaphorin 3A. This is because these 2 genes are characterized as anti-angiogenic by competing with the VEGF, and the upregulation of anti-angiogenic factors could lead to the renal fibrosis. And then I used the progeny to infer the pathway activities, and I found that the VEGF pathway activities were more upregulated across the cells from the renal fibrosis than the normal mice. Lastly, I studied the cell differentiation. For the 4 different integrated data sets, I used the SC-Velo to infer the pseudotime. I found the biologically matched models. 3 of them were the deterministic model, and the last one was the stochastic model. Based on the pseudotime, I got top 20 validated genes of the cell of interest. I reviewed 170 genes across our dataset, and 40 of them have been already described in the literature as driver genes or fibrosis-related genes. I really appreciate the Lafayette group, the Julio group, and the Oliver group. Thank you.